late start. We've had some technical issues, but I think it's been good now for everybody here. So, as you know, the theme of the weekend is superhero, hence why I'm dressed, like a 15-year-old. But we've also got some superhero medical professionals who help us along our way, and one of them is Dr. Stephen Withers, who's generously donated his time today to come and speak. He's a paediatrician and a geneticist, and I think you'll find his talk very informative. So, please welcome Stephen.
Thanks so much everyone for coming along today and coming and being a part of this whole program, because it really is important. I think it's just phenomenally important that people get together, share stories and have a look at what people are doing and how families are working and ideas and concepts that people are using. Some of these are the really valuable things where good information can be shared, and so as a result of that, good things can happen. What I'm going to do today is I'm going to go through what most of you already know. I'll try to go through that in a fairly orderly and straightforward kind of way. One of the things I often say to families is that often families become more of an expert in these conditions than any clinician ever can be. I'm sure all of you have had the experience of saying to a medical professional or a health professional at some point in time, "Oh, and my child has cri du chat," and they go, "What is that? I don't know anything about that. I've never heard of that. Hang on a second, let me just type that into the computer." It's a common and recurring thing. But for you, you are experts. You are experts in the day-to-day, minute-to-minute, hour-to-hour management of this condition. And as a result of that, you have an enormous repository of information that is incredibly valuable. At a scientific and at a research level, what we find is that there's a whole lot of people out there who know all sorts of things at a very scientific level about these conditions, who sometimes have no concept or idea about the practical issues in the practical management of what happens on a day-to-day basis. And I've had that experience myself of talking to people who sometimes I would consider to be world experts in particular conditions, only to have a chat with them and think, you know, you've never sat down and had a chat with a family who's had a child with that condition. You're in the lab and you're looking at all this stuff and you're correlating all this data, but in fact you're not doing the, tell me about how it's going. What's happening on a day-to-day basis? Tell us about that stuff. So look, as I said, I'm going to just go through this. A lot of this is stuff that you've all seen before, but just to sort of reiterate, I do have some stuff just at the end of this, which is some of the, I guess what I would say is some of the new things, and some of the concepts and ideas, and some of this stuff like gene therapy and this sort of stuff. So that's what we're going to get into that. So to quickly go through what all of you know, of course, that you know these are chromosomes. What we know is that in cri du chat there is a deletion, and the deletion occurs in the short arm of chromosome 5. With the result of that being that there's a whole lot of genes that are missing. What do genes do? Genes process things. Genes make things like proteins. Proteins activate things. They switch things on. They switch things off. They increase the production of certain things. They decrease the production of certain things. So what happens is, if you have a deletion there, then what that means is that there is a problem in regulation of a lot of cellular function, and the cells don't work as well as they should. And that's a pretty simple explanation of that. What happens is, and I don't know if people have ever drawn these out for you, this is what we call an ideogram. And what the ideogram is just a drawing of a chromosome that's sort of a bit stylised, a bit cartoon-like, to show, you know, all the bits of it. There's the short bit, the P bit, and of course P is for petite, P is the little bit, and after P comes Q, so Q is the big bit. And what we're talking about is, we're of course talking about 5P, the short arm of chromosome 5. What people have done now is that they've gone through and they've tried to look at all these breakpoints. People talk about breakpoints in the chromosome. Where's the bit missing? Is it right at the end? Is it a little bit further back? Is it a little bit further back again? And when they've looked at this, the region that they're looking at is this part out here. So we've got this top part out here, out from about this point out. And what we do is, if we expand this bit out, these are the features that seem to be correlating with that particular bit missing. And this has been one of the things that people have been trying to do, and this is called, the term for this is correlating the genotype with what we call the phenotype. So the genotype is what the genetics look like, and the phenotype is how a patient presents with a particular condition. So as a result of that, we're able to try and link together and say, gee, if you're missing that particular part of your chromosome 5, that the likely outcome is that you might have an unusual cry, you might have low tone, you might have some developmental delay. And these are the things that they're trying to put together, and people have been working towards trying to sort of nut out what are the particular areas that are important. There's a paper that's come out just in the last week or so that is probably the best overall look at the entire condition, which is available. And I referenced that at the end of this particular talk. And there's nothing in this talk that's not easily findable and distributable. So if people would like a copy of the talk just to pick up that reference, it's a very, very good reference, because of what it is, it's someone who has sat down and they have said, where are we right now? And there's no better time than thinking, where are we right now, than saying that right now, as opposed to 5 years or 10 years ago. What they've been able to do, there's a group in the United States called the 5P- Family Database. So there's a 5P- family group, and they were started in about 1999, and they progressively built up their numbers over a period of time. What they did was, when they first started that group, they put together a questionnaire. And they said, well, what's happening with you? How are you going? Can you tick some boxes and give us a bit of an idea about what's happening? What they've been able to do is to build that database. The data that they've been able to put together for this particular paper involved almost 300 children, some of them adults. But what it's done is it's a fairly comprehensive look at that. And so what we're hoping is that it's from this type of work, of people pooling all this information, that we're able to sort of sort some of these things out, ultimately with the idea of thinking, are there possible therapies that might be available? What do we know about Cri du Chat? We know that it just occurs spontaneously in almost all circumstances. Not all, there are some unusual chromosomal things that can occur. There's a thing called translocation, where chromosomes can be a bit rearranged, and that's one of the other mechanisms by which it can occur. Other things that happen in terms of what are we actually talking about, for most people, there is a loss of the end of the whole chromosome 5 on that short end. So for the vast majority of those, it's a complete loss of that part. So that's called a terminal loss. So that's the complete part, completely gone, and there's no part of that distal chromosome 5P left. For others, there might be just little deletions inside that part of the short arm of the chromosome 5, and then there are some other very rare variations on that. One of the things that's very interesting with this condition is that there are no recurring breakpoints. So in some of these genetic conditions, what we know is that there is a breakpoint that occurs at a certain position. And we go, yeah, almost everyone who has that condition has the same break. And what we find in this is that we don't see that. We don't see a recurrence of a common breakpoint. For reasons completely really unclear to us, we recognise that that short arm of the chromosome 5 carries with it a predisposition to break, and it seems that it can break in a great number of positions, and there's no common theme in that break. So everyone says, well, why is there so much variability in this? You know, there do seem to be people, you know, that people would say, well, gee, you know, it looks like our children have got areas that are sort of pretty much the same. You know, the size of the break point, you know, the piece that's missing seems to be about the same. But everyone says, why is that? Why is that different? And do we have a perfect answer for that? We don't have any perfect answer for that. We wonder about a whole lot of things. We wonder whether there are other genes out there that are doing things and influencing this. We call these modifier genes. Genes that alter or change things, although they're not absolutely critical to some particular area. So that might increase something a bit, decrease it a bit, but it's not a key player in that. There are many other things, other little things, little duplications that can occur in the genes. There's a thing called epigenetics. Epigenetics is an interesting thing, where what we think is that there are probably things that might be inherited from one generation to the next generation. And as a result of those things being inherited, that might modify how something happens in a particular person. And these are all things that are probably playing a role in conditions, for instance, like diabetes in families. Some people have a bit of it, some people have a lot of it, some people get it when they're a bit younger, some people get it when they're a bit older. Why is all that? We think there are all these modifier genes that play a role in that. The environmental factors in all of this, no one's been able to really fully explore that in any particular way as to what we think specially or specifically influences this. It's one of the great problems that we always have, and it's a problem that many people have where people say, oh, why don't we change the diet? Let's use a special diet and that's going to cause some special outcome for us. Why don't we do something a bit differently with, carbohydrates or proteins or something like that, and that will change what happens. And we really don't have the evidence or the information there that strongly supports that. Often people will talk about things in an anecdotal way. Oh, I did this and I felt so much better. I had a large coffee every morning for a week, I felt so much better after that. Did the coffee make me feel better? Oh, it was because I was on holidays. These are the problems that we have. We're trying to get that evidence. It's the evidence that's the really critical thing that we're looking for. People know about the clinical features of the condition, but it can be highly variable. One of the things that we talk about in terms of the features of the condition is often they are features that, as people move into adult life, those features actually are less prominent than they may have been in childhood. So in childhood they may have been a bit more prominent, but in adult life less prominent. Other things that we recognise, people identify things like hearing loss as being one of the common sort of themes that we see, problems with ears, but also vision problems. So vision problems are incredibly common. And we recognise that one of the problems of strabismus, that's where people have squints. So squints are incredibly common. And you may have seen that there's actually an area in the genes that we've sort of looked at there where they think, gee, if you've got that deletion, that probably means that you're probably going to have a problem with squinting. It's just one of those things that we've been able to sort of sort out. Other things that people are very familiar with, of course, issues of poor feeding. This big paper put 44%. I think it's much higher than that. I think it's much higher than 44%. I think that's about 94%, I would have thought, but that's just my anecdotal experience with that, and that I see that as a common and recurring theme. But again, people have looked at that and people looking at different break points without any common theme of the break point. Constipation is a problem, low tone, and low tone is a very common feature. A predisposition to respiratory tract infections and difficulty in moving secretions from the lungs. And scoliosis is considered to be a fairly significant problem, and that's one of the things that people would need to recognise and identify. The complexity of scoliosis is trying to determine treatment, no treatment, what do you do, how do you get the best results from that? Yes?
What's that?
Scoliosis, that's curvature of the spine. So scoliosis is curvature of the spine, and so for children, particularly children who, for instance, might be in wheelchairs and things like that, often from a postural point of view, because they're not standing a lot of the time, then what happens is that they get that idea where they don't need to build the truncal muscles, so the truncal muscles are often a little weaker, so as a result of that they often will end up with a bit of a curve to one side or the other side. That can be corrected with bracing, that could be corrected with surgery, but of course they are very big steps to take. Other things that we know about predisposition to heart trouble, problems with the gut, kidney trouble, sometimes genitourinary problems, behavioural issues. So what we're recognising now is that many of these children would fulfil what we consider to be diagnoses, for instance, attention deficit hyperactivity disorder. So if we were to say, do you think that your child might have that, if they have this condition, we think that the likelihood of that is extremely high. And a lot of parents would certainly agree with that and would see that as something if they get up and going and get particularly busy, hyperactivity can be a very real issue. The other thing which is coming a little bit more on board now is this concept or idea under this umbrella of an autism spectrum disorder. But that can be very difficult, and it can be incredibly difficult diagnosis to make sometimes if you have a child who might have some sort of learning disability. You're trying to figure out how do you put that into the right context. It's a very difficult thing, as I'm sure some of you may appreciate. It becomes very complex. recognising that under the federal government scheme, there's a substantial resource available under the diagnosis of an autism spectrum disorder. And that's becoming a real challenge for us, and I've had many families with children with Down syndrome who bring their Down syndrome children along and saying, here they are, and don't you think they've got an autism spectrum disorder? And you go, oh, I don't know about that, that's a bit of a tricky one, but recognising that there's a potentially big resource, that can be tapped into. That's a very, very difficult problem, not an easy problem, just a difficult problem. Communication, speech delay, of course, is very common, and often they have better receptive language skills, the ability to understand what people are saying versus their ability to express that, and that's a really common theme that we see. We certainly believe that early intervention, and we would always say that early intervention probably as a whole, is likely to lead to better outcomes, but of course that can sometimes have elements of frustration because of course that can be a terrifically expensive resource to access over what might be a very extended period of time. And trying to gauge that right time to actually commence that intervention is sometimes a question that has no perfect answer. And certainly we've seen people who have built up and developed skills really as they've moved into adolescence, having spent childhood where they really have not been communicative. So, as we've said, we've spoken already about early intervention, and we think that these are good things that can come on board. There's certainly the benefit to the early intervention in that those who've had it versus those who haven't had early intervention of some sort. Some of that now is probably a bit more related to people and their ability to access resources over a period of time. There have been people who've lived in relatively isolated communities who've not had the ability to tap into resources. If you live in rural Queensland, then your ability to tap into some of those resources is just about zero, really, because they're just not available in those areas. So, they looked at all sorts of genes, because people wonder, well, is there some special gene that is particularly related to something? And so they found particular genes and they looked at them. These really big numbers here, so if you look at these sort of numbers, what this is the sequencing numbers, and what they're able to do is they're able to go through, when we look at DNA is made up of billions of these little base pairs. And what they can do is they can actually count the number of base pairs to find out where a certain gene appears. So in this one, the top one there, see it goes from about 9 million to about 9.5 million. So what happens is that sort of 500,000 base pairs, that's where that gene is. And it's in that position that we think that if you are missing that as a part of the deletion, that causes potential problems. And you can see all the different parts. So you can see the one here, the ADHD one, that's down in the millions. Some of these others up at 10 million, and so this is what happens. And so we're trying to figure out how all of this works. It's very complicated, what people are working on. So what are the future directions in all of this? Where are people going and what do they think that they can do in the future? The concept or idea is to say, is there some way of having some sort of targeted therapy that is likely to lead to better outcomes? So what you've got to do, you've got to be able to think to yourself, how could I improve the function of something to make it work better? What would be the pathway that we could do that? So what people are looking at is a whole range of different things. This is the idea about saying, well, what can we do to make all of this happen? You might remember that little diagram just from before. So you can see that there's the deleted region in grey and there's the normal region in blue in that the top part of the short arm there. What you've got to try and do, you either have to replace the proteins and the enzymes that were being made by that little area that was deleted, or you need to go to the other chromosome that actually has the ability to make all of these things. And you've got to figure out, can you sort of supercharge it? Can you switch it on and say, I know you're only making a little bit of that, but I need you to make twice as much. Is there some way of kicking that off? And so these are some of the ways that they're looking at doing this. Some of these are mind-numbingly complicated. I think it's, I can't put it in no other way other than say it's mind-numbingly complicated how they think that they are going to get some of those things to work. They are ideas, and they're things that people are looking at and actively trying to think through. The paper that I've referred to, which I've taken most of this information from, talks about this in some detail. I really like the paper because it actually just talks through in a really simple kind of way what they're trying to do. It's trying to give you that idea that if we could do this in some particular way, that would work out really well. But also goes into explaining that there are some complexities in trying to make all of that happen. So, what we know is that they're still trying to figure out so much of this. Participation in research is a very important thing. If people have the opportunity to participate in research, it confers benefit not only to those who participate in the research, but also to those who will follow. And so we have learnt much by people participating in research, even though of course that sometimes is a difficult and complex thing just to be involved in the research. But we know that by sharing this knowledge and information, that can lead to positive benefits. And even like trying to figure out where all those genes are, the common genes that we see. By doing that, that has allowed us now to be able to say, yeah, that's that particular gene involved in it. That would be an interesting gene. Can we upregulate that gene? Can we switch that gene on a bit more? Can we ask that gene to work a little bit harder? And how would we do it? If we'd never been there, we never would have known that. So this is what it's about. There's a lot to learn, there's a lot for us still to do, but people are working on this and people are trying to apply their mind to it. This is the paper. It's a very, very good paper. As I said, quite happy for this little talk to go out on the system and those sorts of things just so that we can pick it up. Sometimes you might have difficulties in getting it. If you do have difficulties in getting that, now I think about it, I'll come up with a way of making sure that everyone can get a copy of it. Okay, so that's probably an even better idea. You'll be able to get the abstract for it, but you probably won't be able to actually get the hard copy of the paper. But it would be something that if you'd like to get the hard copy, I'll figure out how to get that. I'll talk to the team. I will make sure, even if we've got to, even if we've just got to just mail a hard copy out to all the participants, that's probably even a way of doing it. But you know, leave that to me and I'll sort that out. So that was all I was going to talk about now, just to try and sort of say, well, look, you know, this is where we are now. If people have questions, if people wanted to talk about stuff, if people have general questions, really happy to answer those. If people think, okay, well, we've got a bit of a flavour about where we're going and we're up to speed and all that, people will be free to go. But also, if people had special or very specific questions that they wanted to just ask me, if people are happy to hang around, we could work our way systematically. Oh, I see there's one young man there. He thought he had a question.
He's just talking about SeaWorld.
Oh, there we go. My knowledge of SeaWorld is very limited, although it would be a lot of fun.
I met a Ninja Turtle.
Oh, excellent. That's all good news. So as I said, thank you very much, guys.
And more than welcome to come and approach him and have a chat about any specific matters that you might have burning.